talk of today. I'm gonna share with you some results of the study that I mentioned earlier on studying this heat extremophile. This is Death Valley. It's a very beautiful place. If you ever get a chance to go to the US, try going to Death Valley. It's a really like Mars-like place. Used to be occupied by the Timbisha Shoshone tribe. Okay, we know Earth is hot. You've seen this before. You've seen this before. See, this is going to be really fast. This is yeah, the summer temperature of Death Valley in the last 60 years or so. The red is the summer, and then the blue is winter. You can see the temperature really hovers around 50 degrees Celsius, enough to cook red meat. It's called sous vide, 52 degrees Celsius for 2 hours or something like that. Okay, so yes, there are plants that grow in Death Valley. I've told you. Oh, not moving. Oh. Oh, there. This is now I think in the summer. You can see this is pretty much the only plant you see in Death Valley. And it grows pretty big, like a tumbleweed. I told you about these three guys, Ola Bjorkman, Hal Mooney, Joe Berry. This is the science paper from 1972, where they measured photosynthesis in Death Valley. And then this is Karin Prater's work that I'm going to be describing to you mostly. Matt Stada's work on gene family expansion and contraction. I'm going to skip that part. Joanna Feehan is a fairly new postdoc who is asking population genomic questions about Pteudostromia's heat adaptation. So I'm going to end with her story, which is still very early. All of this is very early, I have to say. Joanna is also co-advised by David Lowery and Tom Sharkey. Karin's paper is by archive, but we just submitted our second revision to Current Biology, and so hopefully it will be published soon. So our experimental plan is as follows. We take these plants, we grow them for 30 days, and then we transplant half of them to Death Valley summer condition, and then we measure various things like physiology. We take samples for doing omics, where we did some biochemistry and cell biology. I'm not going to share with you all of the data, a little bit of physiology and a little bit of the transcriptomics. And we went from day 1 to day 18. And the graphs on the right are the temperature and light intensity that we mimicked Death Valley's July conditions. So as a comparator, we use Amaranthus hypochondriacus, which is an underutilized indigenous crop in the Americas. It's actually very high protein seeds that you could use as flour alternative. Anyway, Amaranthus is related to the Pteudostromia, but it's not as heat tolerant. And you can see in a course of 30 days, Pteudostromia really grows well, whereas Amaranthus not very happy at all. And on the graph on the right, it shows you two ecotypes. DV is Death Valley, DP is Dos Palmas, grown under standard and Death Valley conditions. As you can see, around day 7 or the weight, the biomass increases exponentially. They really like being in the hot condition. Now, this is a lot of graphs, I'm sorry, and without a pointer, this is going to be a little painful, but it's very simple. The top 4 graphs are the linear electron transport. Red is Death Valley ecotype, orange is those Palmas ecotype. They both behave very similarly. First panel is 2 days in a standard condition. Second is 2 days in Death Valley condition. The next one is standard condition for 13 days. And then finally, Death Valley condition for 13 days. It's kind of subtle, but I hope you see the x-axis is the temperature going from 30 to 50. And I hope you can see that towards like going about 45 degrees, in the standard conditions, for both 2 and 13 days, they kind of start to go down slightly. Then even as early as 2 days, it kind of stabilizes. So the linear electron transport doesn't decay. So there's adaptation that's observed within a couple of days. We've seen it actually within a day, in fact, of being in that position. When you look at the net CO2 assimilation, you see something similar, more or less. So instead of around 45 degrees or 42 degrees, the CO2 assimilation going down in standard 2 days or 13 days, you can see that they're actually kind of not going down and maintaining. I think if we can get chambers that would be higher, like 50 or 55 or 60 even, it may actually continue to go up. So we saw this adaptation of physiology within 2 days of being in Death Valley, in both ecotypes. Some of you might notice that the Death Valley ecotype actually within 13 days was somewhat better than those counts. And this acclimation of photosynthesis in the Death Valley condition is due to temperature and not light. Because when we put these plants into Death Valley condition, it's both high light and high temperature. This set of panels shows you in blue is the standard condition. The top is the net CO2 assimilation. Bottom is the linear electron transport. Again, the x-axis is the temperature. Okay, the blue lines are standard conditions, and you can see around 42, 45 degrees Celsius. This you can see it much better than these physiological parameters are like performance going down. Okay, in standard condition. When the plants were grown in the standard condition, and you test the physiology at these higher temperatures, they don't do as well. Same thing with light alone, which is shown in green. But if you give it high temperature, which is shown in orange, you start seeing the acclimation. And that acclimation is not really that much different than giving it both high temperature and high light. So this photosynthetic acclimation is really due to temperature and not high light. Okay? And so when we measured sampled RNA in the transcriptomics, we saw a lot of genes go up and down, and all the gene expression analysis in that bioarchive paper. But here I just wanted to show you what happens to photosynthesis of genes. And so the top panel is the C4 photosynthesis cycle, and all of the genes are induced in response to that valley, almost all of the genes. Photosystem 2 genes also go up in response to that valley. Photosystem 1 is not so responsive. That's actually not surprising because photosystem 2 is much more sensitive to heat than photosystem 1. That's a known thing. ATP synthase is also going up. We also saw, not shown here, rubisco activase goes up too in transcript. So I'm going to just kind of wrap that genomics and physiology part right there. And I'm not going to go through the family expansion. So just going to skip through a lot of this slides. We sequenced the genome. We did gene family expansion and contraction. And there's some interesting signals. And we're in the process of writing that up. I wanted to, however, share with you Joanna's work on looking at population genetics of this desert plant. So she started out with asking, where are these plants found? And interestingly enough, all the black dots are kind of the sightings of the plant. And this is shown basically southern parts of California and then a bit of Arizona. It's around Death Valley. And Furnace Creek is basically where the center of that valley is, and the color coding is temperature. So the redder temperature, the higher, the redder the color, the higher the temperature. You can see almost like when you see the Las Vegas, like, oh, it's so hard to, it's like, they really follow the heat, you see? They're only found in the orange and red areas. And this is more visible when you look at herbaria data as well as iNaturalist data. Well, I guess it's not so visible because this doesn't have temperature thing. But anyway, Joanna went around and collected seeds from hundreds of these plants in the southwest of the country. I shouldn't be laughing, but I just thought it was a really crazy idea. But she wanted to do it, and so she's done it. And she wants to do population genetics, this thing. First of all, she found these plants from three different areas. Death Valley population, which is in red, populations that are just surrounding the Death Valley, which is in blue. She calls it perimeter population. And then Tinastromia outside of Death Valley, which is just like in the green. And then these random control coordinates are shown here. And she just wanted to color code these to show the temperature ranges, to show that more or less they're similar in these sightings. But she collected the plants from the red, blue, and green. Okay. Now, and then she did low coverage genome sequencing, about 10x, and found quite a bit of SNPs, about 42 million. And she saw that there was actually a SNP, one SNP every 200 base pairs. So even though, so basically she saw that there's enough genetic variation to do this population genetics thing. And so then she asked, okay, how diverse is this set of population that she found? And this is basically FST, or fixation index, that shows you how similar or different these individuals are genetically. And it basically shows you that the red and the blue are very similar, as you can see in the FST index. The red and blue guys from that valley are pretty different from those that are found in the outside of that valley, but they're not that different. So she's found in a different study that there doesn't seem to be a lot of population structure, which means another sort of supporting evidence that she can do GWAS or something like GWAS. Okay. So she next asked how much heterozygosity there is, and it looks like there's some heterozygosity, but based on this number here of observed heterozygosity, it suggests that this is a selfing plant, and that's indicative of the flower structure too. They were kind of complete flowers and stuff. Anecdotally, we were never able to really get the seeds in our greenhouses, except that when we were kicked out because of COVID, we were told to leave the lab in the morning. This was when COVID started. And then, we had to leave the lab by 5 p.m. And so we had to leave all our plants in the greenhouses, and they all died. And so we were able to come back about 4 months later in California, and we saw seeds in Tinastromia. So all we needed to do was just like stop watering them and wait for about 4 months. But now we are able to get seeds thanks to COVID. Yes. So now, with all of this information about genetic diversity, she wanted to set up an experiment to do the GWAS, but she didn't have enough individuals really to do GWAS because she got seeds from about 40 sites, I think 120 or so plants. So what she wanted to do was something like bulk segregate analysis type of thing, where she would give these plants really extreme heat to kill off about 90% of the population, and then sequence the genomes of the surviving 10% and compare them to either randomly selected populations or the ones that were more sensitive or something like this. Okay, so in a scheme like this, that she would give them really high temperature, like 60 degrees Celsius. The pink represents the plants that have died, and the green represents the plants that survive. Okay. So she actually did this experiment a lot of times and got a kind of protocol that worked. And this was to treat them at 60 degrees for 8 hours, I think. And this is the actual data that she got. She got about 10% surviving and 90% or so dying. So she's in the process of sequencing them, but in the meantime, she saw that, she asked, like, where are the, survivors and the dying ones? And she saw that they were actually all across those three sites that I told you about. Within that valley, a perimeter of that valley, and just outside. So again, it shows you that maybe this is a single population. So she's in the process of doing that, GWAS, but in the meantime, she saw, because she was trying to measure this phenotype with a camera inside that crazy Death Valley chamber. And we bought this like infrared camera, and she saw that there was actually an interesting difference with the leaf temperature. And of course, like, it's known that plants can cool themselves like we do through transpiration. And so she thought, okay, well, is there something that's associated with survival and leaf temperature reduction? And so she actually mapped that trait. And so this is a PCA plot that's showing survival in 3 different clusters. This has nothing to do with those geographic sites now. Sorry for using the same colors. But on the left, on the graph on the right, it shows you this is survival probability on the y-axis, and then x-axis time to death, in days. And so you can see that the cluster 1, the peak one over there, survives much better than the blue or the green. Okay. And so she then asked, like, is there any temperature difference among these groups of plants? This is a hard thing to see, but this is a temperature of these plants as a function of time during her treatment regime. And I'm going to blow it up for you because it's hard to see. In the first 2 hours of 60 degrees, there's no difference among those 3 populations. Purple is the soil temperature. But in the last 6 hours of being at 60 degrees, you can see that the cluster 1, which was the better survivor guys, its temperature is much lower than the other 2. And this is actually more visible when you look at the last 2 hours versus last 6 hours. Okay, so they're of being 60 degrees. So there's something going on with these survivors having cooler temperature and maybe more efficient transpiration. She asked, like, is there any correlation between leaf size and survival? And she found that there's no correlation. Okay, so that's kind of where we are. She's kind of fitting the temperature thing with an energy budget model that's been developed by Tom Sharkey and others a long time ago to ask if it's really like preparation, if there are anything else that may be causing this. And so we're still having a lot of fun doing different aspects of examination of this plant. And we are looking at identifying candidate genes that might be involved in heat adaptation from various methods. And we're also very interested in starting to test these candidate genes in related crops like sugar beet or quinoa or amaranth. And yeah, that's kind of where we are right now. I'm going to end by thanking Corinne mostly, Joanna and Matt and the rest of the collaborators and members of our lab. This is actually quite old, doesn't have Barno in it. Next time I give a talk, Barno will be on this one. Thank you, everybody.
Thank you so much.
Save you a lot of time. Questions?
Yeah, thanks. But for the next talk, so I have a question in regard of the plants that you have analyzed in the Death Valley. So did you ever check general morphology of the leaves or going deeper, checking chloroplast, granulocyte, something like that?
Yeah, I have another talk on that.
Okay.
It's in the bioarchive paper. So first of all, at the leaf level, there's a lot of trichomes. And the trichome density increases as a part of the activation process. And when you go look at the chloroplast, it's very interesting. So the leaves get smaller, and this because the cells get smaller. And so the density of chloroplast and mitochondria increases. And the positioning of chloroplast and mitochondria, they come together. That's another sort of observation that we made. And also in the bundle sheath of chloroplast, that belly time, we see about 20% of the time, this chloroplast change their shape to be like cup shaped. And the cup thing is it's like a reverse cup, facing the vascular. So this is the leaf surface. And it's kind of like, pumping the vascular.
Yeah.
So those are the initial observations that we've made, but there's a lot of very interesting cell biology and morphological and anatomical changes going on.
I would.
Yeah, and we're starting to look at the roots as well, in the collaboration with Salk Institute folks. There's a lot of suberin me, and then, yeah, I think the roots are going to be as interesting as the shoots because they have to like get this transpiration stream going. And there isn't a lot of water over there. And so the way they grow really fast isn't by growing really deep. They don't make tap roots, they make very shallow roots. But the soil of that valley is kind of clay-like. And in the summer, there's this thing called summer monsoon in that valley. So there's a rain actually for about 3 days, 3 to 4 days each summer. And it gets, because it's so hot, it comes in and then it gets like sealed by the heat and then the clayness of the soil. And then the plants basically like really vacuum up all of the water and then transpire like crazy, grow like crazy, and then they flower after the heat is over.
Thank you for very interesting. I was enjoying it so much because it reminds me almost the same project on Elosia with Brazilian genetics as extreme resistance. And actually, my question is regarding this temperature things because in our previous labs, so they found that there are some microbiome communities, and even they developed some kind of microbiome, microbiological culture, which with the treatment with these cultures and plants can regulate the temperature during this extreme heat conditions, and with certain microbiome environment, temperature leaves somehow lower. Did you check this?
Do you mean in the phyllosphere or in the rhizosphere?
So I'm not sure because how they applied this microbiome, but actually in the research project, so we checked both an aerial part of the plant and the root microbiome, but it was some kind of it looked like microbiome somehow would contribute to this temperature.
Yeah. So we haven't done that, but we have some data, like the transcriptome data and also the genome data, and go back with the lens of specifically looking for the sequences that couldn't be mapped to the genome, and see like what we got. Because these were certainly not sterilely grown plants, but they were also not grown in that valley in situ. So we haven't really collected stage samples that were collected from that valley. But I think it's a really interesting question.
But they were collected from different locations, as far as I understand.
Yeah, we collected seeds mainly. And so we didn't really collect tissue. And the tissue we collected, I mean, like we didn't really do transcriptomics with them. So yeah, I mean, I think it's a really interesting question, and I think it would be really cool to see what's there. And the answer